Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about the different properties of the enzyme in the course Enzyme Science and Technology. And what we have discussed in the previous module is that we can actually be able to study the interaction between the enzyme and the substrate, or enzyme and the different type of ligand what are interacting with the enzyme with the help of different types of techniques. So in this context, what we have discussed about the chromatography techniques, whether the chromatography techniques are going to be used for measuring the change in shape or size of the molecule, or whether it can be used for measuring the or detecting the different types of charges onto the enzyme structures. And in addition to that, we have discussed about the couple of spectroscopy techniques. We have discussed about the isothermal titration colorimetry. And then in the previous lecture, if you recall, we have also discussed about the surface plasmon resonance technique. So all of these techniques are very easy to perform, and they are actually being, and majority of these techniques are label free techniques, so they can be very easily or economical in terms of usage. So with this, we have understood that the enzymes are interacting with the substrate, and enzymes are catalyzing the different types of reactions. So if you recall, we have discussed about the different types of enzyme catalyzed reactions. We have discussed about the catabolic reactions, and we have also discussed about the anabolic reactions. So when an enzyme is processing a substrate, what it is doing is enzyme is reacting with the substrate, okay, and then it is actually forming the transition state, which is called as enzyme substrate complex. And then this transition state is getting converted into another transition state, which is called as the enzyme product transition state, right? And from the enzyme product, you are actually going to have the uh, release of enzyme, and you are actually going to have the generation of product, right? And this enzyme will again go into the cyclic manner, and it will actually go and receive the another molecule of substrate. Now what how we can be able to measure the enzyme activity, right? So the ability of an enzyme to process the number of substrate molecule is actually going to be a direct measurement of the activity, right? So number of molecules. So if you want to measure the enzyme activity, you have two options. Either you can be able to count how many number of molecules of substrate are being used, or you can be able to say how many number of molecules of products are being formed, right? So either you can be able to measure the formation of the product, or you can be able to measure the depletion of the substrate, and then only you can be able to measure the enzyme activity, right? Now, if you want to do this, you are actually going to have a very well documented or well managed infrastructure, so that you can be able to measure the substrate or the product. And if you want to do that, you are actually should have the exclusive properties related to either the substrate or the product, right? So you should have the exclusive properties. Um what is mean by the exclusive property is that these exclusive properties will not be present, or will not be present in any other species throughout this particular cycle, which means if you are measuring the substrate, it should not have that you are measuring the substrate, but at the same time the product is also very much similar and is also giving you the similar reactions. And that's why if you want to do the enzyme activity, you require a, or you actually have to standardize a system which is called as enzyme assay. And in the enzyme assay, you are actually going to use the different types of criteria or different types of conditions in which you are actually going to either measure the depletion of the substrate, or you are actually going to measure the formation of substrates. So in today's lecture, we are actually going to discuss about some of these aspects and how you can be able to set up the enzyme assay to measure the enzyme activity. So as the name suggests, the enzyme assay is a assay where you are either going to measure the depletion of the substrate or the generation of the product. So the enzyme's activity can be determined by determining the rate of product formation or the substrate used in the enzyme catalysis reactions. Many enzyme activity can be determined by the multiple assay procedures. The choice of assay depends on the cost, the availability of appropriate equipment and the reagent, and the level of sensitivity required. So this is very important parameter what you are supposed to consider when you are trying to develop an enzyme assay for measuring the enzyme activity. So it depends on the multiple procedures. It depends on the availability of the equipments, such as the if you have the spectrophotometer or not, right? So if you have the spectrophotometer, then only you can be able to, or you should actually set up a assay where you are actually going to have the change in the visible region, right? If you have the fluorimeter, you can actually be able to set up the or design the enzyme assay accordingly. So depending on the equipment, you are actually going to set up the enzyme assays. Then you also require the different types of reagents. Suppose, for example, I am trying to develop a protease assay. So actually I require the different types of proteins, right? Because that is going to be the substrate, right? So whether I should have that protein or not, then only I can be able to use or develop a protease assay, right? And then what is the level of sensitivity required? If you require an assay which actually can work in the micromolar range or millimolar range, then or versus whether you are looking for a sensitivity where you are actually going to measure the differences between the substrate and the product in the range of nanomolar range and so on. So depending on the sensitivity, the cost is also will actually going to be, go up. So all these are, and the most important thing is cost actually, because when you are trying to develop the enzyme assays, the cost is one of the very important parameters, because that is actually going to decide how many number of reactions you can be able to perform, right? So if you are actually going to run an assay where the cost is $10, for example. So if you are actually going to have an assay which has $10 cost for every assay you are going to do, like this means you are actually going to use approximately rupees 1000 for performing single reactions, right? So this is going to be very costly. So that also is actually going to be a limiting factor to go and adopt that particular type of enzyme assay, because if you can measure the enzyme activity even with the cheaper options, maybe you may have to compromise with the sensitivity and other things, but you will actually at the end you will get the answer, right? Then it is essential that you ensure that the any assay procedure which gives a true measure of the activity of an enzyme. So that is very important, that when you develop the assay, it should actually give you the right readouts, and it should give you the real assay or real procedure. So in this procedure, you should actually first figure out what are you are going to measure, and whether there will be a cross reactivity, because as I said in the past also, right, in the previous slide also, that the substrate and products are not very different, but they are different in terms of some of the properties. For example, if you are measuring an assay where your glucose is getting converted into glucose 6-phosphate, uh, these two are very different, except that you are actually going to have a phosphate group which is attached to this substrate, this product actually, right? So you are actually can only use this as a probe to measure, right? So that's how you should have a very good procedure in which you should not get misguided and you are measuring something which may not be reflecting the activity of an enzyme. All the potential problems may be avoided by carefully experimental design and adequate control. So when you are trying to develop the enzyme assays, you are going to have two reactions. One, reaction 1, which is actually going to be blank, or I will say control, right? Where you are going to have everything except you can actually be able to remove the enzyme. Then you are going to have the reaction 2, where you are actually, or I will say this is actually the test reactions. Sometimes you may also have the multiple types of blanks. For example, you can have the control for enzyme, you can also have the control for substrate, you can also have the control for other things, right? So if that is the case, you should be little more careful, and you should actually be able to set all these reactions. Then only you can be able to measure the enzyme activity. Some seemingly abnormal behavior in enzyme assay can provide valuable information about the properties of enzyme being studied. So when you perform the enzyme assay, it is actually going to say how the substrate is going to be processed and how it is actually going to convert that into the product. And this is actually a very, very important information. It actually going to tell you the behavior of that particular enzyme, because if you do a subtle change in pH or subtle change in temperature or pressure or other kind of environmental parameters, it is possible that the same enzyme probably maybe start processing the different types of substrate. It may start generating the different types of product and so on. So that would be a very, very abnormal behavior of the enzyme assays, and that is going to be also give you the very, very critical informations. Now, when you try to develop the enzyme assays, you also going to see the different types of behavior of these enzymes, right? Enzyme assays. So these behavior is actually going to give you a lot of information about the enzyme and its interaction with the substrate and how it is actually processing the substrate to actually giving you the product. So let's see what are the different types of behavior of the assay. So when you are going to study the behavior of the assay, you can actually be able to expect that the reaction is actually going to process in these three way, right? It can either be a curve A, it can follow a curve B, or it actually can follow a curve C. In either of these cases, what will happen is that when you are actually going to run an enzyme assay for time versus product, right? And if suppose you are measuring the product, what will happen is that initially it is actually going to be a linear, and then it is actually going to saturate. Okay, so this is actually the non-linear regions, and this is the linear region, right? And it actually can follow different path. It may actually it can be linear, or it can be reaching to this point much faster, and it can actually be able to not be able to reach this. Okay, so that's how it is going to have the three different types of curves. So when you do the reaction progress curve, or you are going to monitor the enzyme assay, and you are going to see how the enzyme is actually processing the substrate, and it is actually going to give you the product. And if you are measuring the product, not the depletion of substrate, what you are going to see is you're going to see the multiple types of curves, right? So initially the time course is linear. So what you see here is that in this area, the time course is linear. For example, in this one also up to this, it's linear actually, and it started that plateau actually. So this is the non-linear region. This is in fact, there's no change in product, right? However, the rate of product formation begin to slow over time and linearity is lost. There are several reasons for the loss of linearity. Okay. So these are the three type of model which can be possible. So a model progress curve of an enzyme catalyzed reaction. A, when you are going to get A, when the enzyme has the high value of both Km and the Vmax. So if I think you should not be worried about the Km and Vmax, but these are the assays. What you are going to see is when the both the Km and as well as the Vmax is going to be very high. Then the curve B you are going to get when the Km and the Vmax values are lower. So these are the curve what you're going to get. And the curve C is the reaction where the reaction rate is slowing because the enzyme is unstable and it loses activity at a constant rate, with a complete inactivation having occurred before all the substrate has been converted. So this is the reaction where you are expecting an enzyme which is not very stable. So it's not stable. And what happened is that the concentration of enzyme is also changing. So here what happened is that the concentration of enzyme is changing over the time. Okay. So you can imagine if that happens, then it is actually going to keep processing the lower and lower and lower substrates, and that's how the enzyme, instead of increase in enzyme, instead of increase in product formation, there will be a decrease in product formation, and that's how this is not going to be the good. So you can actually have the three scenarios A, B, and C. And why there is a loss of linearity? So there are multiple reasons why there is a loss of linearity in this particular system, or I will say why the system actually reaches to the saturation. So the number one reason is that you are actually going to have the substrate depletions, right? Because enzyme, what enzyme is doing is enzyme is processing the substrate and it is generating the product, right? So if there will be a substrate depletion, or if there will be a decrease in substrate concentration, right? Because this is equilibrium, right? So if the decrease in substrate concentration, enzyme will not have the adequate substrate so that the binding should occur, and that's how it is actually going to be get converted into the product. Then it's also going to be because there is a change in equilibrium, right? So because there is a because all these are under equilibrium, so product is getting converted, or it's actually reversible actually. So at equilibrium, the concentration of so initial concentrations are high, but when the system reaches to the equilibrium, the forward reaction and reverse reactions are equal, and as a result, there you will see that it is actually going to adopt the plateau. For example, in this case, right, forward reaction and the reverse reaction is actually going to be equal, and that's how it will not go down. So it will go linearly to some extent, and then it will actually going to attain the equilibrium, and that's how it is actually going to have the plateau, or there will be a loss of linearity. The number three is the product inhibitions. So you can have the product inhibitions, and this is actually also going to be called as feedback inhibition, right? So when you have the some product is formed, right? So this product could be inhibitor for the enzyme, and that's how initially it may not be having significant impact, but when a lot of product is formed, it is going to inhibit the enzyme, and that's how it is actually going to make the curve linear, or not. There will be loss of linearity. Then the fourth point is the instability of one of the component of the enzyme assays. So when you are actually going to use the different types of components, some component could be temperature sensitive, for example, right? So when you and majority of these assays are being performed at 37 degrees Celsius, right? Because that is the temperature at which most of the enzymes are showing the activity, optimal activity, right? So but the components could be temperature sensitive. For example, if you are using the ATP, right? So ATP is good, ATP is stable molecule, but at 37 degrees Celsius, the auto oxidation of the ATP to give you the ADP plus PI is very high, right? And as a result, there will be a degradation of ATP, and that's how it may actually going to not available for enzyme for catalyzing the reactions, and that's how it can actually be able to give you the non-linearity, or the product is stop going up, right? Then you can also have the time dependent inhibitions. So there is a time dependent inhibition is also like product inhibition, that after some time the system is reaching to the equilibrium, or after some time the product is formed which is actually going to have the inhibitions. Then sometime you can also have this kind of behavior where you can actually losing the linearity, because there is a artifacts in the system. So it is actually artificially showing the plateau, but it is actually not. And the number six is change in the assay conditions. So this is very important, right? You are actually going to have the change in pH or temperature. So when you are actually going to have that, for example, in some enzymes, what happen is that when the enzymes are processing the substrate, for example, at this site, when you are actually doing it initially, right? So in the initial reaction, you have set the pH, everything right. But when the product is forming, and suppose the product is acidic in nature, right? So if the product is forming which is, acidic, right? So that is going to slowly going to start affecting the pH. Okay. In other cases, sometime you are actually going to have the assay where the assays are endothermic, right? So when the product is forming, it is actually also affecting the temperature. Although majority of these assays are always being done in the incubators, or they are actually being done in the water bath. So you always maintain the regular temperature, but the pH is always can be modulated, right? Because if you are forming some acid, like for example, if you are generating the lactic acid, right? So lactic acid is a acid, right? So it is actually going to impact the pH of the media, and as a result, the initially the pH is 7.4, but when the lactic acid is started producing, the pH could actually come down to 5, right? For example, so if the enzyme is optimally active at 7.4, but it may not be active at 5, and that's how you might be able to see that there is a loss of linearity. So these are some of the reason why you could be able to see a loss of linearity. And because of this reason, majority of the people, what they do is they are actually using this part of the curve only for measuring the enzyme activity. So they don't allow, or they don't use the whole curve for measuring the enzyme activity, because this is the curve which is very, very reliable. And compared to this, because at this point you are actually reaching to saturation, and when you are reaching to a saturation, there are many things what can actually go wrong. You can actually have the substrate depletions, you can actually have the product inhibition, and so on. And you can also have the sometime artifacts which are actually also possible. So that's why this is the initial curve where you are actually going to do a lot of measurements, and majority of the enzyme kinetics people are using this particular curve. What are the different features of these initial measurements are? So when you do the initial measurements, what you're going to do is, initially the rate of product formation is linear with respect to time, but as there are many reasons for the loss of linearity over time, to avoid such complexity, is it important to measure the initial rates. Okay, this means you are actually going to measure only in the initial part of the curve, right? Because that is going to be linear, and you know that a linear part is actually going to give you the accurate information. Frequently, the linear portion of an assay is sufficiently extended to allow the initial rate to be accurately estimated simply by drawing a tangent or taking a first derivative or the early part of the progressive curve. Okay, so this is very easy, because when you are doing the initial curve, it can actually be able to extend it, and then, or that's how, or you can actually be able to derive the equations, and that's how you can be able to use that part of the curve to calculate the different properties or different kinetic parameters of the enzyme. The concentration of the enzyme is reducing to prolong the linearity of the progressive curve, whereas the loss of linearity is very rapid. This also increases the sensitivity of the assay. So when you are doing the initial rate measurements, you are also going to increase the sensitivity of the assay, because in this particular range, in this particular curve, the uh, the assay is very sensitive. Suppose you add any kind of inhibitor, this actually is going to get affected. It's possible that the saturation point may not get affected, but this portion is going to be get affected. It has frequently been assumed that the limiting measurement of reaction rate to a period in which less than 10 to 20% of the total substrate consumed will provide a true the initial rate measurements. Okay. Starting an assay by introducing one of the component and ensuring adequate mixing can lead to the considerable uncertainty about the precise time and the reaction had begun. So one of the major issue with the initial measurement is that you are not very sure about when you started the reaction, because when you are going to add one component, the last component, then the reaction is going to start. But the precise time when the reaction had begun is very difficult to say, right? So sufficient time must be allowed to adjust the initial conditions, such as the linearity is maintained for enough time to measure the initial rate. So it's not only important that you are actually going to make the initial measurement. It's also important that you allow the enzyme to get stabilized, because initially the enzyme is actually going to take time to recognize the substrate. Then it is also going to start catalyzing. It's not like it started jumping on all the substrate and then started eating them, right, or started converting them. So it's actually going to be a molecular interactions, and initially the molecular interactions are going to be slow, and then it's actually going to catalyze. So that's why it is not important that you do measurement in the as early as possible. You can actually let the enzyme to get stabilized, get started reactions, and then you actually can measure. Okay. And then you can have the entire progress curve must be taken into account when measuring the complex enzyme assays, where substrate depletion and equilibrium is not the sole reason for the loss of linearity in the progress curve. Now, you can see that even if you are doing the measurement in the initial curve, there are many issues related to even the initial measurement as well. And the worst issue is the burst and the lag in the progress curve. So what you can see here is that I am showing you the two curve, where the later part of the curve, right, this part of the curve is different from this part of the curve, right. This part of the curve is actually showing a phenomena which is called as burst, right. This means the enzyme is starting very rapidly, whereas in this case, what you see here is that this part of the curve is very different from this part of the curve. So this part is actually showing a lag phase, which means the enzyme is taking very long time actually to recognize the substrate and actually going to catalyze the reactions. So if you are measuring the reactions in the initial time curve, or very initial time curve, then you are actually going to make a mistake, because if it is a burst conditions, you are actually going to do the overestimations. Okay, and if it is a lag kind of enzyme, you are actually going to make the underestimations. So before the linear phase of the reaction, in some cases, there is either a burst or lag in the product formation. And the potential reason includes is inadequate temperature control. So sometimes what happen is that there is a temperature is not isothermic, so it's actually going either be very high, whereas in the case of burst, or it could be very low in the case of in the case of lag. So that actually is also going to control the activity of the enzyme. Then sometime when you are doing the reactions, the reaction components may not be soluble into the buffers, and they will be keep settling during the reaction. So what happen is that, suppose this is a test tube, right, and I have taken the enzyme. When I have taken the enzyme, all the components, so what happen is that some of the components are particulate, right. So what happen is they will go. So after some time, what will happen is they will actually going to be settled down at the bottom, right. And once the enzyme is present here, right, so enzyme is present in the solution, but they are some of these particles, which are also important for the reactions, are settling down at the corners. So that's how they were actually going to change the reaction rates, and they may actually induce the lag or burst. Then sometimes we have the slow detector response, which means your measuring devices are also having the fault. So they will be actually either measuring the very quickly, or they were taking time to measure, and because of that, enzyme is catalyzing its reactions, but you are measuring very slowly. So that's why you can actually have this particular type of curve. Sometimes you have the slow dissociation of a reversible inhibitor or the activator. So in that case, if you have slow inhibitions, it may actually be able to, have the lag phase, a longer lag phase, or if you have the slow dissociation of activator, you may have the burst. And that's why these are the two phenomena which actually going to make the initial measurements wrong. Then you can have the pre-steady state transients or relief of the substrate inhibition or activations. Then you can also sometimes have the activation of the product. So this is very, very interesting, because when you have the product which is actually being working as an activator. So what happen is that in the lag phase, what happen is that when the enzyme is converting a substrate into the product, and then the product is actually working as an activator. So initially the curve is actually going to be slow, but as soon as some amount of product is formed, it is actually going to activate the enzyme, and that's how it is actually going to activate the activity of the enzyme. Then you can also have the substrate interconvergence, which means the substrate are actually going to be get converted to each other, and that's how they are also going to affect the burst or the lag phase in the progress curve. So the time course of the enzyme catalyzed reaction showing the burst or the lag phase before the steady state rate is obtained. This is all about the enzyme. So in an enzyme, when you are actually going to do enzyme catalyzed reaction, what you are going to do is you are actually going to have these, right? This means you're going to have the product and the enzyme is being formed, right? And if I set up the reactions, what I'm going to do is I'm going to set the blank reactions, right? So in a blank reaction, what I'm going to do is I'm going to take the buffer, right, where I'm going to catalyze, for example, tris pH 7.4, right? And I'm going to add the any kind of salt or additives. For example, in this case, I am supposed keeping it 100 millimolar NaCl. So it's actually going to give me provide some kind of biological environment. And I'm going to add the some amount of substrate. So for example, if this is a ATPase, for example, so I'm going to add like ATP, which is 10 millimolar, right? And that's it, okay? And then I'm going to add the substrate. So I'm going to add the substrate like, for example, glucose, okay? So 100 millimolar, for example, okay? So I'm going to add the glucose. So I'm going to add the ATP and everything. Remember that I have not added the enzyme. So this is actually going to be a blank reaction. So imagine that I got an OD after 10 minutes, for example, okay? So if I did the incubations, after 10 minutes, I am going to get a OD of 0.4. So this is the 0.4 absorbance, okay? So this is the OD values. Now I am going to do a test reaction, okay? So test reactions, I am going to have everything. I am going to have the buffer, I am going to have the salt. So everything I am going to take from the blank reactions. I am going to have the ATP, and I am also going to have the glucose, right? And at the end, what I'm going to do is I'm going to add the enzyme. So for example, I have added 10 units of enzyme. So, and then I will start the reactions. And after 10 minutes, I am going to have an OD of 0.8, for example. Okay. This means if I want to know what is the enzyme catalyzed reactions OD, so what is the change in OD, right? So what I'm going to do is I'm going to do the test OD minus blank OD. Okay, this means 0.8 minus 0.4, and that is the 0.4 OD what is being changed when the enzyme was added, right? And this is what you are actually going to calculate after every time points, like 1 minute, 2 minutes, 5 minutes, like that, okay? And what you will see is that this matter, this value is also going to be changed. For example, if I do it after 1 minute, I may have a value of 0.05. If I do the after 5 minutes, I will have this value of 0.1, and so on. So this value is very, very important, because this is actually going to change your final values. Okay, and that's why this value is actually going to be contributed by the blank rates or blank reactions, right? And the blank reactions are very important in a enzyme assays. So blank assays or blank rates. So sometimes the apparent rate of reaction is observed even in the absence of one of the component of complete assay, which are called as, and those are called as blank rates, right? And blank rates are very important because they are actually going to be decide what would be your final rates of the enzyme. Because if the blank rates are very high, for example, if I am getting a OD of 0.8, 0.9, then your the sensitivity is actually will be very low because the blank rates are very high. So it is important to ensure that the determined rates from a particular assay is only due specific reaction is only due to the specific enzyme catalyzed reactions. So potential cause for the blank rates include settling of the particles, precipitations, contamination of one of the component of the assay mixture, adsorption to the assay vessels, and non-enzymatic reactions. So this is the non-enzymatic reaction, the blank what I am talking about, or some of the contaminating enzymes. So that are also going to be responsible. How you're going to correct about the blank rates, right? In many cases, the true rate of the enzyme catalyzed reaction can be obtained simply by subtracting the blank rate given in the suitable incomplete mixture from the rate obtained with the full assay. So that's why I said, when you subtract the 0.4 from 0.8, that is what you are going to get. So this is actually the corrected OD values for this particular time points, for example, 10 minutes, right? And that's how you can be able to overcome the blank rates, because ideally, when I'm not added the enzyme, ideally there should be no formation of product, but in some of these cases, you are actually going to see some of the enzyme which is going to be formed. This method assumes that the blank rate is an artifact unrelated to the activity of enzyme under study. That is, continuous linearity for the duration of assay, and that it remains constant throughout the assay. In some cases where these assumptions are correct, failing to subtract the blank rate will result in the visible kinetic anomalies. Then if the blank rates occurs, failing to subtract it results in a plot of initial velocity versus enzyme concentration that does not pass to the origin but exhibit finite activity as a zero enzyme concentrations. If the apparent blank rate is due to the particle settling, subtracting the initial blank rate from the initial rate obtained after starting the reaction may be sufficient. However, such rates are normally irregular, and it is preferable to wait for the blank rate to decline and the assay to stabilize before measuring the rates. In a more complicated system in which it is inappropriate to subtract an apparent blank rate can occur when the enzyme can catalyze the decomposition of one of the substrate alone, but the presence of the second substrate suppresses these reactions. So the dependence of the initial rate on the enzyme concentration. So what you're going to see here is that the initial reactions are, or initial measurements actually depends on the concentration of the enzyme, and they are also going to be very sensitive for these blank rates, okay, because the blank rates actually can reduce the OD values. So what you see here is that I have shown you how the enzyme concentration is actually going to change the rate of the product formations. So in the line B, what you see, so it's three conditions, A, B, and C. Okay, so B is shows the expected dependent line, which means it is actually going to show you a linearity at 45-degree angle. Okay, so this is actually going to be ideal. Whereas A and C showed the possible result from the incorrect treatment of the blank rate. In line A, the blank rate occurred in the absence of enzyme has not been subtracted. So this is actually the ideal rates what you're going to measure in the absence or in the when there is no the blank rates. But if you have the blank rates and if you don't subtract those blank rates, then you are actually going to expect a curve which is look like as the curve given in the A. Whereas the C, a blank rate occurs in the absence of one of the substrate, but it is suppressed in the full assay has been subtracted. So this is actually the under representation. This is the over representations of the same enzyme assays because the blank rates are affecting that. So the effect of the enzyme concentration on the enzyme kinetics or the product formation can be of two types. It can be directly proportionality. So in most cases, the initial velocity of the reaction is directly proportional to the concentration of the enzyme. The graph of initial velocity against total enzyme concentration will be straight line passing through the origin. So that is what I have shown you, right? This is actually going to be like this. So initial rates are very, very sensitive for the concentration of the enzyme. And this is not true in all cases of derivation from this linearity, maybe due to change in assay condition, and with the time like change in pH or the ionic strength, it is important to take this factor into considerations. So you can have the two different types of effects when you are going to see the effect of enzyme on the initial rate measurements. It can have the upward curvature, or you can also have the downward curvatures. So let's see what is the upward curvature. So when you are actually going to see how the enzyme concentration is going to induce the upward curvature, so there are two reasons for this type of behavior. So what you're going to see is that when you are actually increasing the enzyme, what you see is that there is an increase in upward curvature, right? Which means the velocity is increasing. And so these are the initial rates, okay? So there are two reasons for this kind of behavior. The presence of a small amount of a irreversible inhibitor of the enzyme in the assay mixture, and the presence of dissociable activator in the enzyme solution. So upward curving dependency of initial velocity on the enzyme concentration. So curve A shows the normal expected relationship, right? So this is the curve A, which is going to show a linearity, right? Linearity between the enzyme concentration versus velocity of the enzyme, velocity of the reactions. Whereas the B represent the case where there is a irreversible inhibitor contaminating the assay mixture. So B is the time place where when you are actually going to have the irreversible inhibitors, and because of that there will be a delay in initial kinetics, and that's how it is actually going to show you the linearity afterwards. And then the C is the show the possible behavior if there were a reversible inhibitor activator present in the enzyme preparation. So when you are actually isolating the enzyme, it also contains one of the activator along with it. So when this activator is present, it is actually going to activate the enzyme activity over the course of time, and that's why it is actually going to show you an upward curve. Then you can also have the downward curvatures. So there are three common scenarios that can result in a curvature curve with a downward curvature where the reaction rate appears to be reach maximum at a high concentration rate. The detection method may become rate limiting at the high rate concentrations. So these are the downward curvature depend on the initial velocity of the concentration. So what you are going to have is when you are keep increasing the enzyme concentrations, initially it will be linear, but later on it is actually going to be downwards, okay? And one of the major reason is that the detectors or the detection system what you are going to use for these kind of assays may not be, will be the rate limiting, which means they will not be able to measure the substrate or the product what is being formed, which means when the enzyme is reacting with substrate and it is forming the product, the concentration of product is very high. So concentration of product is very high, and as a result it is actually reaching to a saturation level, and because of that it is actually going to show you a downward curvature. Then failure to measure the true initial rate of the action because of the responses of these instruments and other kind of thing, you will not be able to do the real initial measurements of the reactions. And the presence of a dissociable inhibitor in the enzyme solution. So sometimes you may have some kind of inhibitors, and that may also getting dissociated, and that's how it is actually going to show you the downward curvature. Now, whether you are getting the upward curvature or whether you are getting the downward curvature, the enzyme is actually going to show you activity, right? which means because of this activity, the enzyme is going to convert the substrate into the product. Now, the question is how you are going to express this activity and how you can be able to use that information for determining the other kinds of parameters. So how you can be able to express this activity, because you can have the enzyme from the multiple sources, you can have an enzyme from animal source, you can have the enzyme from plant sources and so on. So how you will compare which enzyme is more active and which enzyme is less active and so on. So there is a universal activity, what is, or activity definition, that is what required to compare the enzymes from the different sources, even the same enzyme from the different sources. So the expression of the enzyme activity. So one of the unit is unit and the specific activity. That is the universally very popular way of expressing the enzyme activity. So what is the one unit of enzyme? So the most used quantity to express the activity of enzyme is the unit of enzyme, sometimes referred to as the international unit or the enzyme unit. What is the one unit of enzyme? It is defined as the catalyzing the conversion of 1 micromole substrate or the formation of 1 micromole product in 1 minute. The specific activity of an enzyme preparation is the number of units per milligram of proteins. If the relative molecular mass of an enzyme is known, the activity can be expressed as the molecular activity, which is defined as the number of units per micromole of enzyme. In other words, the number of moles of product formed or substrate used per mole of enzyme per minute, and that will actually going to give you the molecular activity. Because an enzyme molecules may contain more than one active site, this may not correspond to the number of mole substrate converted per enzyme active site per unit. If the number of active site per mole is known, the activity can be expressed as the catalytic center activity, which corresponds to the moles of substrate used or product formed per catalytic center per minute. Or so this is the so the enzyme activity can be expressed in three ways. One is you can express it in the terms of unit, you can express it in terms of specific activity, which is equivalent to unit divided by milligrams of protein, or you can actually be able to express in terms of the catalytic center activity. So catalytic center activity, where you are actually going to say that unit of enzyme divided by the number of active sites. Okay, so number of active site of the group. Okay, and either of these three way you can actually be able to express the enzyme activity. But recently the people have also adopted the new uh, activity units for measuring the enzyme activity. So one of the that unit is cattle. Although the unit of enzyme activity and the quantity derived from it proven to be most useful, the International Union of Biochemistry and Nomenclature Commission (IUPAC) has recommended the use of cattle, abbreviated to CAT, as an alternative. So 1 cattle correspond to the conversion of 1 mole of substrate per second. Thus, it is inconveniently large quantity compared to unit. So the relationship between the cattles and the units are 1 cattle is equivalent to 60 moles per minute, or 6 into 10 to power 7 units. So you can actually be able to use this unit, which is a recent unit, and the relationship between the cattle and unit is that 1 cattle is equivalent to 6 into 10 to the power 7 units, or 1 unit is equivalent to 16.67 nanocattles. However, in terms of the molar or catalytic center activity, the cattle is not such a large quantity, and it is consistent with the general expression of the rate constant in the per second. While you are doing this activity measurement, stoichiometry is very important. So it is important to keep in mind the stoichiometry of the reaction when expressing the activity of the enzyme. Sometimes the enzyme catalyzed reaction 2 moles of the same substrate, for example, the 2 ADP, right? So in that case, you might have to, change the concentrations accordingly. So the activity will be twice as large as it is expressed in terms of ADP utilizes than if it is expressed in terms of the formation of either product. When expressing an enzyme activity, it is critical to specify the substrate or the product as well as the stoichiometry. Now, there are multiple way or multiple conditions what you have to consider before you are going to design an enzyme assays and you are going to use that for measuring the activity. So conditions for the activity measurements. So although the velocity or the enzyme concentration is a useful constant for comparison, it also be constant under the specific conditions. For example, the pH, temperature, and the substrate concentrations. Because many of these parameters are actually going to change the enzyme activity. And that's why it is important that you should also define that at what pH, at what temperature, and at what initial concentration of the substrate you are getting this activity. Because it's not that the enzyme is actually going to show you the same activity even if you are doing the reaction at 4 degree or 27 degree or something like that. So that's why the enzyme activity you are actually going to show, but you also have to show the activity conditions in which what temperature you have used or what pH you have measured and all that. Because if you even if you do like pH measurements and if you do the activity at a pH where the enzyme is not very active, it's not going to show you the activity. The temperature of 30 degrees Celsius has become widely used as a comparative standard, but in some cases a more physiological temperature may be preferable. There is no particular pH and substrate concentration is recommended. Often the optimal values are preferred for the specific case, but to get the activity of enzyme in vivo conditions, physiological pH should be used. Now, you have we have discussed so what we have discussed about the different types of enzyme activities. We have measured how we can be able to or express the enzyme activity. We have also discussed how the different parameters are actually going to impact the activity measurements, the initial rate constant and all that. Now we should discuss how and different ways in which you can be able to measure the enzyme activity. So type of enzyme assays. So enzyme assays could be of two types. Either they will be direct continuous assay or they could be indirect continuous assay. So although many enzyme catalyzed reaction product produce change in the property of reactant that are relatively easy to measure directly and continuously, others do not, necessitating the use of an indirect method that involves some additional treatment of the reaction mixture. In some cases, such direct measurement can be used to continuously monitor the progress of reaction, but in their cases, reaction must be stopped before further treatment of the assay mixture can be determined. So you will see that when we are going to discuss some of the assay mixtures and when we are going to discuss about some of the reaction conditions, you will see that how some of the assays are continuous direct measurements and some are continuous indirect measurements. So one is the continuous direct measurements. So any difference in the substrate properties and the product can be used directly measured, can be used to provide the basis for direct assays. Direct assay means the enzyme is making a substrate and it is forming the product, right? And you can easily measure this product directly, which means either the product is colored or the substrate is colored, and you can actually be able to measure how much the substrate left, right? So either of these cases you can actually be able to do direct measurements. Individual activity has been measured using fluorescence, pH, optical rotations, conductivity, enthalpy, viscosity, or the volume of the reaction mixture. Direct continuous assay are always preferred because they allow the observation of progress curve, which simplifies the estimation of the initial rates and allow the detection of any anomalous behavior as long as the sensitivity is sufficiently high and the procedure does not impose undesirable limitations on the assay condition that can be used. So it is very easy and it is straightforward. That straightforward, you see that the product is forming, and that's how what you can do is you can just plot the product with time, okay? And that's how you can be able to calculate the initial rate constant. You can actually be able to use that for calculating the different types of other kinds of kinetic parameters, whether the Km, Kcat, and all that kind of thing. So that is desirable, but many of the cases what happen is that neither the product is very good in terms of unique properties, or substrate is also not good in terms of unique properties, so that you can use that to measure, and there is an interference. So when you are having that kind of assay, you are actually going to use the indirect methods to measure the assays. So indirect assays or indirect measures. So indirect, you can have discontinuous indirect assays, you can have the continuous indirect assays. So discontinuous assay, where you are actually going to have like enzyme plus substrate forming a product, and then you are actually going to recover, or you can actually be able to purify this product, and then you are going to put it into another reactions, okay, and that's how it is actually going to be the discontinuous indirect assay. So these assays, also known as sampling assay, involve stopping the reaction for a predetermined amount of time and treating the reaction mixture to separate a product for analysis, or produce a change in the property of one of the substrate or product that can then be measured. Radiochemical assays are the examples of these kind of assays. So for example, you can have the ATP plus luciferin plus oxygen, and it is actually going to give you the oxyluciferin, okay. And this is the one of the assay which actually can be used to measure the ATP, for example. So if suppose this is the assay what I am doing, right, glucose plus ATP giving glucose 6-phosphate plus ADP. So if I want to measure this, it is very difficult. So what I am going to do is I am going to recover the ATP, and I am going to put that into this assay, and that's how it can be used for measuring the ATP concentrations, because the oxyluciferin is actually going to give you the light, and that light can be measured. By measuring the light emission in the presence of firefly luciferase, the formation or the disappearance of ATP can be determined. Similarly, you can have the NADPH FMN, and it is actually going to give you the NAD plus. And here also you are going to have the FNOH2 is formed, right, and that can be put into this reaction, and that's how it is actually going to produce the light, and that light can be measured. So this is actually going to be a indirect method, because after some time you have to take out these reactions, and then you have to measure the ATP. You cannot do simultaneously. Then we can have the continuous indirect assays. So continuous indirect method, where you are actually going to have the indirect measurements, which means the substrate is actually going to, product is actually going to measured, but you don't have to aliquot. You can actually be able to add something, and then the product is actually going to form the P prime, and that is actually going to have the exclusive property which can be measured. So this type of assay entail forming the manipulations required to detect product formation or product remaining within the assay mixture in such a way that the change can be tracked continuously as it occurs. Such assay should allow for the determination of the progress curve in a single assay, making them the less prone to errors caused by the sample manipulation required in a discontinuous assay. So continuous assays are much better, because they are actually going to reduce, first of all, they are actually going to make the things faster, right, because you can actually be able to add everything, and then it's actually going to give you the direct measurements. The second is it is actually going to be less prone to the artifacts, because in this particular system, when you are aliquoting the reactions mixtures, and you are actually taking out some amount, it is actually not going, it's going to interfere with the enzyme kinetics. In the assay mixture, reagent that react with one of the reaction product to form a detectable compound can be included. To yield the valid results, the detection reaction must be so fast that the reaction catalyzed reaction is always rate limiting, so that the rate determined component to the activity of enzyme under assay. This is very important parameters when you are trying to design the continuous indirect assays. Then we have the coupled assays. So coupled assays means you are, this is the examples of coupled assay, where the glucose is getting converted into glucose 6-phosphate with the enzyme of the hexokinase. So if we want to measure the activity of hexokinase, we have two options. Either we couple the formation of ATP to some system, or we can actually be able to couple the glucose 6-phosphate to another system, right? So we can actually put the glucose 6-phosphate to glucose 6-phosphate dehydrogenase, and that's how it is actually going to show me depletion of NAD plus, right? And that can be monitored both ways. That can be monitored with the UV visible spectrophotometer, or it can also be monitored with the help of the fluorescence, because these molecules are fluorescent molecules, so they can actually be able to show you the fluorescence. So the most common type of assay uses one or more additional enzyme to catalyze a reaction of one of the product to yield a product compound that can be detected directly. This type of assay is known as coupled assay, and the auxiliary enzyme used are often referred to as a coupling enzyme. Now, when you want to do a coupling reactions, okay, it is very important that the activity of this enzyme should be on a higher side compared to this enzyme, okay, so that the conversion of this substrate, conversion of this product to the this product should be faster, because if this enzyme is fast and this enzyme is slow, then there will be an accumulation of the product, and that's why it is actually not going to give you the direct measurement, or it is not going to give you the real image or real situation of the product concentrations. So it is very important that when you choose the coupled assays, or when you try to develop the coupled assay, the coupling partners, the second enzyme, which is actually going to be called as coupled enzyme, should have a higher enzymatic activity, so that there should be no accumulation of the product from the first reactions. So this is the reaction 1, and this is the reaction 2, right? And reaction 2 should be fast, so that there should be no accumulation of glucose 6-phosphate. So this is all about the enzyme assays. So what we have discussed about the basics of the enzymes. So we have discussed about what are the different enzyme assay system, what you can use, what are the precautions, and what are the factors which are actually going to impact the enzyme assays. And then we also discuss about the effect of enzyme concentration on the assay system. And the purpose of this discussion is that so that you should be very careful with the concentration of the enzyme into the particular enzyme assay system. And then we also discuss about the role of baseline or the blank rates, and how it is actually going to impact the enzyme activity. And lastly, we have also discussed about the units of the enzyme activity, and also we have discussed about the different types of enzyme assays setup, what you can actually be able to use to measure the enzyme activity. So with this, I would like to conclude my lecture here. And if you want to study, or if you want to discuss some more about this aspect what we have discussed today, you can actually be able to refer this particular book. And I have taken the content from this book, so it will be easy for you to follow the content. So with this, I would like to conclude my lecture here. Thank you.